Thank you. Thanks for the very kind introduction, and thank you all. I know it's the end of the day, I know it's been a long day, so very much appreciate you hanging around to hear about Argenica. So I'll just give you a quick snapshot of, I guess, the company as a whole. We are a clinical stage drug development company, so that means we're in the clinic, we're about to test our drug in stroke patients. And we're developing novel neuroprotective drugs. So these are drugs that are able to protect brain cells following brain injury. So the first indication we're going after is stroke. That's the indication that we have in the clinic at the moment. But we've got some really exciting data, preclinical data coming up with traumatic brain injury, Alzheimer's disease, and a condition in newborn infants known as hypoxic ischemic encephalopathy. Say that fast at 3 o'clock in the afternoon. So I'll talk about that a bit, but I really want to focus on the stroke indication in this presentation. So currently there are no drugs on the market that are able to protect brain cells following a stroke, following HIE, following a TBI. So we're going after some huge unmet medical needs, and we're one of only a handful of companies that are actually in the clinic testing these novel neuroprotective drugs. What makes our drug unique is that it doesn't work on a single mechanism. It's actually what we call plurifunctional. So it's working on multiple mechanisms in that cell death pathway, and that is really important for neurological drugs. Neurological conditions are complex. There's lots of things going on. So having one drug that targets just one thing is never going to work. So we're really excited by the plurifunctional nature, and this is starting to really pique the interest of pharmaceutical companies. Which is exciting. It's exciting when they actually start knocking on your door as opposed to the other way around. So we listed the company in June 21. We were a preclinical company at that stage. So in that 2 years, we've managed to scale up our drug manufacturing. We can manufacture it to what's known as GMP grade. So that's the grade that you need for commercial drug development. We've conducted all our safety studies preclinically, all our toxicology studies preclinically. We've tested the drug in healthy human volunteers in a phase 1 study, and we've submitted our ethics for our phase 2. So we've been pretty busy over the last 2 years, but hopefully it gives you the confidence that we've got a really fantastic team that's able to deliver on milestones. So in doing that, we've managed to increase our share price from IPO by 90% and generated significant value for shareholders. So a couple of key metrics here. So we've got just over 9 mil cash in the bank at the moment, and another couple of mil coming through from our R&D tax in the next couple of months. We've also managed to get around $4 million of non-dilutive grant funding, and that's really to fund the preclinical applications. So the cash that we raise in the company is purely sort of earmarked for the stroke phase 2 trial, and we do access this non-dilutive grant funding for our other indications, which is fantastic that we can do that. And sitting at the moment at a market cap of around 36 mil. Cheap. So a couple of key takeaways from this slide. So the lead drug candidate is Arg007. It is a peptide, which is essentially a small protein drug. It's made up of 18 arginines. The drug itself was discovered over 10 years ago at the Perrin Neuroscience Research Institute and the University of Western Australia, both based in Perth. So it's had over a decade of preclinical work done on it. So this is not a flash in the pan, quick kind of story. We're about 12 years into the drug development process. So we're actually at the really, the critical end of that drug development process. We've got over 25 peer-reviewed publications. We just announced another one on Tuesday, looking at implications for the drug in Parkinson's disease. So a huge amount of de-risking work that's already gone on in terms of the efficacy of this drug in a number of these preclinical animal models. And now, again, we've shown that it's actually safe to deliver to humans and well tolerated. So I'll just quickly explain how it works in ischemic stroke. So someone that has an ischemic stroke, it's caused by a clot in a blood vessel in the brain. There's two types of strokes. It can either be ischemic, so the clot, or a hemorrhagic stroke, which is a bleed on the brain. about 85% of all strokes are ischemic, so caused by this clot in the brain. And the clot can either arise in the brain or it can arise somewhere else in the body and travel to the brain. And what that clot does is it stops the blood flow to that part of the brain. The brain cells can last about 3 seconds without blood flow. So they need the oxygen that the blood delivers to be able to survive. And once brain cells die, they're dead, they're gone, they don't regenerate. And because brain cells all talk to each other, what you get when you have that reduction in blood flow and one brain cell dying is it causes the next one to die and then the next one to die. So you get this proliferation or cascading out of cell death in the brain. And so there's a saying in stroke that time is brain. So about 1.9 million brain cells die every minute that treatment is delayed. And so obviously this results in really dire functional outcomes if you have this delay. So what we're trying to do with this drug is shut down or hibernate those brain cells. So what all our preclinical data shows is that this drug is actually able to stop that cascading out of cell death. So we can't do anything about that initial bit of cell death, but as soon as our drug's delivered, it shuts down these neurons, these brain cells, it hibernates them and protects them from all that nasty stuff that happens in the brain when you've got that lack of oxygen. And then our data shows that these brain cells, these neurons, come back to life after about 12 hours and they restore normal function, which is obviously really important. So you've got this about 12-hour window where you can get that patient to hospital, remove the clot, and hopefully completely reduce that amount of brain cell death. So ideally what we want to do is to be able to give this drug in the ambulance. That's sort of the ideal scenario. A phase 2 trial is designed slightly differently. I'll come to that in a minute. But essentially, if we can give our drug, stop that brain cell death, buy that patient time to get that clot removed, either through a clot-dissolving drug or a procedure called a thrombectomy, where they mechanically remove the clot, then they're going to have better functional outcome. So this is some of our preclinical data. This is in a non-human primate model and really convincingly shows how our drug is reducing that total amount of brain cell death by 66% at 24 hours post-stroke. But that's also lasting, so once blood flow is restored, where you can often get a secondary wave of brain cell death, that we're still having a 70% reduction in brain injury, which is really quite phenomenal. So really encouraging data to date. We've got that non-human primate data showing that huge reduction in brain cell death. We also know that this drug can be administered to hemorrhagic stroke patients and it won't exacerbate bleeding. So the current treatment, the clot-dissolving drug for ischemic stroke, if that's administered to hemorrhagic stroke patients, they bleed out. It exacerbates the bleeding. Our drug doesn't do that. So we can give it regardless of the type of stroke and therefore before diagnosis. We also know that it can be administered concurrently with the clot-dissolving drug. So we have published data to show that. And that's really, really important when you're looking at standard of care. You know, you want to be able to deliver your new drug with whatever the current standard of care is. So what really sets this drug apart? It's all these things that I've spoken about. So having that multiple mechanism of action, really important in stroke, because there's a number of different pathways of how those cells die. We've got huge amounts of preclinical efficacy data to really de-risk this drug asset. We also know the drug is a cell-penetrating peptide. For drugs you're trying to get into the brain, that's really important. You need to know that it actually crosses the blood-brain barrier and gets to the cells where it's needed. And as I said, doesn't exacerbate bleeding and can be delivered with clot-dissolving drugs. So I probably don't need to explain to you that the market opportunity that we're going after is massive. Current treatment of stroke is $183 billion. We have done a lot of work looking at pricing of this drug and the number of ischemic stroke patients actually presenting to emergency departments just in the US. So if we just take the US alone, delivering it to 600,000 people at $10,000 an administration, we're talking about a pretty huge market where there's currently no drugs actually on this market at the moment. So as I said, we've been really busy. We've ticked huge amounts of milestones to get into phase 2. It's not an easy process to take a drug from preclinical into a phase 2 trial, especially getting over that hurdle of a phase 1, showing that the drug is actually safe when you're administering it to humans. And now we've submitted our ethics to seek approval for that phase 2 trial. So if I just touch on the phase 2 trial quickly. So this trial will be done only in Australian hospitals, and it will be done in Australian hospitals that are able to perform thrombectomy. So that's the mechanical removal of the clot. And the reason that we're only administering it to patients that are eligible for thrombectomy is because you get this secondary wave of cell death from the thrombectomy procedure itself. So when you remove the clot, you get this huge rush of blood back into the brain, and it actually, the shearing forces of that rushing blood back into the brain causes secondary cell death. And 50% of patients that have this thrombectomy procedure actually do worse off, and they believe it's because of the reperfusion injury. You see it as well in organ transplants, this idea of reperfusion injury. So we're giving ourselves the best chance to also see efficacy because of this reperfusion injury. So we'll have a couple of hospitals in Melbourne that will be on this trial. So we've got the Alfred Hospital, and we're also bringing on Sunshine Hospital as well, who interestingly don't do thrombectomy, but will be delivering our drug there and then bringing them into thrombectomy centres into the CBD. The whole purpose of the trial really is to look at safety, so looking at safety in ischemic stroke patients, but we will be measuring infarct volume, or the total amount of brain cell death, via imaging as a secondary endpoint, as well as collecting a whole bunch of functional recovery data. So this is absolutely critical for our company. This is really where we've got that proof of concept that our drug works in stroke patients, and this data coming out is going to be the thing that will allow us to do a deal with a pharma company. So we should be dosing first patients by the end of this calendar year, early next year. And we have an amazing team that is leading this trial. I might quickly skip over this, but they're fantastic. So Tina and Jane, both based in Melbourne, and really taking a lead role in running this trial. Because I just want to quickly touch on the pipeline. So the phase 2 trial is blinded, so we won't get data out. So we obviously need news flow as we go along and starting to progress some of these other indications. So traumatic brain injury, obviously massive market. Currently no treatments available for traumatic brain injury. When we talk about traumatic brain injury, mild TBI, which is concussion, there is going to have to be a therapy. So it's all well and good having protocols in place, absolutely, and that reduces the inflammation, but we need something that's going to protect against the initial injury. So what you see that happens in the brain when someone suffers a TBI, you get inflammation, but you get this calcium influx, really similar to what you see in stroke. So we've recently announced preclinical data to show that our drug is having a significant impact in protecting brain cells following TBI. And we've got a federal government grant to progress that work into concussion. Alzheimer's, so what happens in neurodegenerative conditions, Alzheimer's, Parkinson's disease, is you get an accumulation of proteins, beta amyloid protein, tau protein, and alpha-synuclein. And it's this aggregation of these proteins that sits on your brain and causes the dementia. So we've now got really nice data to show our drug reduces the aggregation of beta amyloid, it reduces the aggregation alpha-synuclein, and we're hoping it will also reduce the aggregation of tau. My time's up, isn't it? And we've also looking at this hypoxic ischemic encephalopathy. So this is when the cord is around the neck in newborn infants, the mother has preeclampsia, and that baby suffers a lack of oxygen or a hypoxia. And so we've got, again, really great preclinical data, so we'll progress that. And you're standing up, and these are the investment highlights. And thank you very much, and the presentation, and I'm outside. Thank you.